Yeah,
Yeah,
I thought that, in the
yeah, sir.
meanwhile, we'll utilize this time to start presentation on our, society. Society is, registered officially. Our objective is to like pick up our capabilities into one forum and then develop our profession into a better profession and like a respected profession. And we value everyone's contribution and appreciate major or minor contribution from anyone that is coming to our society, so that we can contribute better for our society. We also invite wholeheartedly all colleagues who would like to serve for our society, so that our profession becomes a more lovable and fashionable society. And we hope all MD pharmacologists will accept our invitation, and like we also request everyone to support, so that we will be able to serve better for our profession. So this is a composition of our society. You can see that people coming from all over India are there. Dr. Kaushal from Ludhiana, Dr. Pooja from Kolkata, she is in Bangalore and Hyderabad. Dr. Melvin is from Chennai, and Dr. Lakshmi Narayan is from Ballari. Dr. Malini is from Bangalore. Dr. Poolan Sharma from Chandigarh. Dr. Akash from Belgavi, and then Dr. Divya from Bangalore, Dr. Jildi Basu from Kolkata, and Dr. Sushma, she is from Shimoga. So she is currently in USA. So like this, we have society who are not located in one place, and they are all widespread all over India. And we look forward for having people coming from all the different parts of India to join us, and then strengthen the society. And we have also formed four councils which can act as like arms and legs for serving the society. So we have a CME council headed by Dr. Sandeep Kaushal and vice chaired by Dr. Pooja Rai. So they will be responsible for executing and taking decision on CMEs. And we have research council headed by Dr. Poolan Sharma. He is DM clinical pharmacology from Chandigarh, and he will be we have a lot of programs related to research council, and so he will be leading them. And Dr. Melvin George, he has already initiated the discussion on journal and newsletter related activities, and he will be responsible, and we have the whole array of people in that. And then myself, I am leading the team of media and public relationship, and I have the team with me for doing the justice for the organization. So this is our organogram. In the basic stage, we have established our office bearers, and now we are in the stage. We have also formed the functioning councils. We will also start working on the like core committees for various states and student council, and so remaining activities. So this is the organogram. So you can go through it in leisure. What are the aims, like how why this society is required? So MPS is a registered society, and as per the acts of Society Act, and mission is to provide a platform for MD pharmacologists for innovating themselves to serve the community better. So currently we don't have a proper forum which can represent the MD pharmacologists. So we would like to we are providing a platform for that purpose. And vision is to build the MD pharmacologist profession as the most respected and most innovative one, with the combination of all the scientific and technological advances. So what are our objectives? Objective is to quality of education, and improve the quality of education by modifying the syllabus and providing the time to time suggestions to the governing bodies. And employment opportunities, support the newcomers and anyone who is in need. So that we are going to do. And training and development of the professionals using the various platforms like this, and also CMEs and conferences, workshops, or whatever possible. And business ownership. So currently, like MD pharmacologists are not owners of the business. Like it's a very interesting area. So we never like we don't see very large number of people getting into that. We could see few people like Dr. Malini and Dr. Amit Dang. So these are the people who are already there in the business front. But we can, we would like to support the MD pharmacologists to become entrepreneurs for a period of time and give them all the required skills, education, or mentorship, whatever possible. So we'll try to do that. And specialty labs, we would like to develop specialty labs where, our MD pharmacologists can go there and get the skills so that they can become a better person over a period of time. So then multidisciplinary approach, we would like to collaborate with, both national and international associations, also governments, so that we can bring a lot of resources for the profession so that we can serve better. Collaboration, as I told you, with government organizations and international universities. And in addition, we would like to take the leadership by providing, like the standard operating procedures for all the people, whoever would like to, come into the sector. We would like to support them so that, like in the beginning, if we can give them, for to become a bud, then I'm sure they will become a flower over a period of time. So we would like to do that type of support and own journal and newsletters so that, we can communicate to the society that, we are this profession, we have these goals, and this is what we have achieved. So if we can do that, then people will start recognizing as a better profession, and it's a good profession and as a preferred profession. So then establish, like boards for reviewing the, data, like data reviewing committee or ethics committees, and also like pharmacovigilance centers, so that, we can support the government and also the research and development of various sectors, and support the pharmaceutical companies by taking up the investigator initiated trials and conduct all over India, and even if possible required, we will collaborate with the international universities or the companies, so that we will be able to do a top-notch research and support the publications. And then, over a period of time, who knows, we would like to we will, in such a way that we would like to have the labs which can develop the molecules, and then we can try right from the molecule to market. So we can do that, and we can support in large, like bringing the value for the society, value for the profession, and value for individuals. So this is what is this thing. And memberships, we have already started the memberships, and membership forms are available. So we offer life membership, and then student membership and honorary memberships. So details are in the our bylaws, and also in the forms. And the forms also contains the instructions for filling the form. So please go through it, and whoever is interested, please try to contact us, and we will support you. This is our contact address. Thank you very much.
Hello. Okay, good afternoon, everyone. Welcome to your society, that is Medical Pharmacology Society. Dr. Murthy has already explained about various aspects of the society, why it was formed, who are the members, what is the role of the society. And I would like to thank Dr. Murthy for giving me this wonderful opportunity for presenting the very first webinar of the society. It is also a part of the academics. So we are here for a webinar to discuss various strategies to develop inhibitors of motif-mediated protein-protein interactions as drug leads. So next slide. Yeah, we will be covering the topic under the following heads. First, the introduction, then various methods to discover the motif, various strategies that are required for optimization of the motif that is discovered, various strategies that are required for delivery of the motif, and finally the summary. Next slide, sir. So before proceeding further, we should know the various jargons that are going to be used during this whole webinar. So motif is nothing but a distinctive sequence on a protein or a DNA having a three-dimensional structure that allows binding interactions to occur. So here, since we are mainly primarily focused on protein-protein interactions, so the motif is going to refer only the distinctive sequence on a protein and not the DNA. When the peptides are the compounds consisting of two or more amino acids linked in a chain. The peptides can further be classified as oligopeptides and polypeptides depending on number of amino acids that are linked by peptide bond. Plasmids, these are the small DNA molecules within cells that is physically separated from the chromosomal DNA and can replicate it independently on its own. Patch, these are the viruses that infest and replicate within a bacteria. Intin, these are the segment on a protein that is able to excise itself and join remaining portions, that is extin, with peptide bonds. These intin, considering an example like alphabets A, B, and C, considering example alphabet A, B, and C, which can be used to represent the segments of the protein. The alphabet B can act as a intine while and it can actually excise itself and join together the other segment, that is extend A and C by a peptide bond. Deep sequencing. It is a sequencing a genomic region multiple of times, even hundreds or even thousands of times. Peptide arrays. These are collection of peptides displayed on a solid surface, usually a glass or plastic chip. Epitope, it is a part of an antigen that is recognized by immune system like antibodies, B cells, T cells, while the paratope is nothing but the part of antibody that recognizes an epitope and binds it. Canonical amino acids, these the naturally occurring 20 proteogenic amino acids, which are also called as standard amino acids, that are encoded directly by triplet codon in the genetic code. Non-canonical amino acids. These are mainly non-proteogenic amino acids, but only two proteogenic amino acids are known, that is 21st amino acid selenocysteine and 22nd amino acid pyranolysine, which are encoded via variant codons like stop codon. Chaperone. These are the proteins that assist covalent folding or unfolding and assembly or disassembly of other macromolecular structures. Plasmid. These are plasmids that contains an F1 origin of replication from an F1 phage. Origin of replication is a particular segment in a genome at which replication is initiated, while F1 is a phage derived from origin of replication that allows replication and packaging of single-stranded DNA into the phage particle. Now coming to the introduction, protein-protein interactions are the fundamental basis of almost all cellular processes. It is estimated that there are approximately a total of 650,000 interactions in human proteome. Thousands of short peptides have been identified that play crucial role, such as hormones, neurotransmitters, growth factors, ion channel ligands, and anti-infectives and cellular signals. Natural peptides often represent an excellent starting point for drug development, and compared with traditional small molecules, peptides are less toxic, highly specific, and are easy to synthesize. More than 50 peptide drugs are already approved and are being marketed in the drugs and include lipirolin, pegnicetide, gozalin, octreotide, and fubertide. Lipirolin and gozalin are gonadotropin releasing hormone analogs and are used to treat symptoms of prostate cancer, endometriosis, breast cancer, uterine fibroid, and precocious puberty. While octreotide is a somatostatin analog and is a more potent inhibitor than a somatostatin of growth hormone, glucagon, and insulin. It is used for the treatment of growth hormone producing tumors like example is acromegaly and gigantism. Pagnicetide is a erythropoietin receptor agonist and is used in the anemia associated with chronic kidney disease in patients who are on dialysis. The disadvantage of these the peptide drugs which are formed by the inhibition of protein-protein interaction is since they are unable to cross the cell membrane passively, they mostly work extracellularly. They have got low bioavailability and have got poor pharmacokinetics, that is, they have got a low resistance to hydrolysis by proteases enzymes, they have got faster elimination, and I have got a lability, that is, they are readily or continuously undergoing physical or biochemical changes. So various strategies have been developed to overcome these disadvantages and improve peptide drug efficacy, and these include chemical modification and encapsulation, which will be discussed in subsequent parts of the webinar. Despite these challenges, a number of intracellular peptide drugs reaching their later stages of clinical trial is increasing, like clingetide, which is indicated for glioblastoma, is in the second phase of clinical trial. Now coming to the first topic of the webinar, that is discovery of motif. The ideal starting point in peptide drug design is crystal structure of the protein which is implicated in a disease state bound to a short sequence fragment of a protein partner. This structural information can then be used as a framework for optimization of the natural interactions by approaches such as rational design and medicinal chemistry. In reality, this scenario is relatively rare, with a very good exception that is an example of MDM2 P53 complex. The p53 is a guardian of human genome, and it conserves stability of the cell by preventing genome mutation. While MDM2 is an oncogene, and it is a negative regulator of p53 tumor suppressor, and thus by targeting MDM2 p53 complex, various drugs can be formed. While in most cases, information on the complex or the peptide motif or both is lacking. So, several methodologies have been developed to screen polypeptide libraries for ligands targeting selectively expressed receptors. Coming to the very first methodology, that is fast display. It is by far the most utilized high throughput methodology to study protein peptide interactions. It is based on the fast display is based on displaying peptides on the coat of bacteriophage by expressing peptides as fusion to capsid protein. So here you can see from the diagram the elongated portions and the elongated portions are phages, and the beads that are attached in the blue color like blue, pink, green, purple, these are the peptide which are bound to the phages and form the phage library. And the plate that has round things, these are the bait proteins which are or the targets which have been placed in the plate. So DNA library coding all the peptide sequences or the combinatorial library of randomized segment is cloned into a fragment vector to create a phage library. Next, the binding clones are enriched through several rounds of screening against an immobilized bait protein that is in the plate. Finally, the tight bound peptides are identified by rapid sequence analysis, and these are subjected to wash by solvent, and the unbound phages are washed away, while the specifically bound phages are eluted with an excess of a known ligand or the target, or by lowering the pH. Well, elution is nothing but removal or separation of one material from another, especially with the help of a solvent. These phages are, after 3 rounds, individual clones are isolated and sequenced. The choice of phage and the coat protein are important factors in modulating the display valency of the fusion protein on the phage particle, where the choice of the phage may be M13, T7, or lambda phage. This technique allows the construction of libraries with a high diversity, that is up to 10 to the power 10 variants, at a relatively low cost and time expenditure. And main advantage is that you need not have the prior knowledge of the target structure. The applications of fast display, it is used to identify new protein peptide interactions, conducting affinity maturation of known motifs, screening of short linear peptides with random sequence in vitro, to select for tissue specific peptides by in vivo selection rounds in animal and human cells and tissues, and to identify D-amino peptide ligands. The drawback of fast display is that it is limited to the 20 naturally occurring amino acids. It is not well suited for capturing interactions that rely on post-transcriptional modification, and the motif prediction are not always accurate and may be unspecific. Pegnicetide, as I have already explained, it is a erythropoietin receptor agonist used for the anemia in patients of chronic renal failure, which who are on dialysis. Is an example of the peptide which has been discovered by fast display. Yeast surface display is an alternative to fast display. In this display method, peptides can be expressed on the surface of the cell in fusion with outer membrane coat or on bacterial flagella. Here you can see in the figure, in the yeast display, each cell carries plasmid DNA, and the DNA in the plasmid is a nothing but genotype. As in the you can see the small circles in the yeast cell, the plasmid is being shown, that is of the size around 5 micrometer. The genotype is nothing but a DNA carried by the yeast, which code for a peptide that is displayed on the yeast cell surface. As you can see, the phenotype in the larger circle in the red color, phenotype is shown in the protein displayed on the yeast cell surface. This is displayed on the yeast cell surface as a fusion with AGA2P subunit of the mating protein A agglutinin. Cells are then incubated with target coated magnetic beads or fluorescently labeled soluble targets. Cells displaying mutants that bind the target are isolated through magnetic activation cell sorting or fluorescent activated cell sorting. The advantages of yeast surface display over the bacteria-based technology is that cases in which cell surface and secreted proteins require endoplasmic reticulum specific post-translational processing for efficient folding and activity, which cannot be done by other display methods, and it allows the scientist to obtain information on non-binding clones. Well, the limitation of yeast surface display it is that the libraries are usually 100 to 1000 times smaller than that of fast display libraries. Now coming to the cell-free systems, there are two types of cell-free systems, ribosome display and mRNA display. First, we will talk about ribosome display. In the ribosome display, these screen polypeptides in vitro with large libraries based on the formation of stable peptide ribosome mRNA complexes in a cell-free system. As you can see from the figure, the DNA encoding the combinatorial libraries, that is combination of peptides, is transcribed in vitro to an mRNA fused to a spacious sequence lacking a stop codon. So the mRNA that is transcribed does not have a stop codon. What the stop codon normally does, it triggers the dissociation of elements which are involved in the transcription. That is ribosome, peptide, and mRNA. And this complex can be dissociated. But due to the absence of stop codon in the ribosome display, it consequently ensures that normal process does not take place during the cell-free translation and thus allow the formation of the complex of a peptide together with the encoding mRNA and the ribosome. These tertiary complexes are then exposed to the cell immobilized target molecules. As you can see, immobilized antigens are complexes are exposed to immobilized antigens. The non-binding molecules of the library are washed away while binding peptides remain. From these bound complexes, the mRNA are isolated, reverse transcribed, and PCR amplified for further manipulation. And these include repeated cycles of selection to enrich peptides with high affinity, protein expression, and deep sequences for analysis. So we have already discussed these things. Now, second part is the mRNA display. mRNA is just same as a ribosomal display, but the differences are that the formation of covalent link between the nucleotide template and the expressed peptide via puromycin linkage. A puromycin is added, and as you can see, so one slide back, as you can see over here, puromycin is linked with a mRNA via a DNA linker, and this is in vitro ligation. The puromycin is added at the 3' end of the mRNA template, covalently links the mRNA and the polypeptide when it reaches the ribosome. Puromycin is nothing but the analog of the 3' end of the tyrosyl tRNA, with a part of its structure mimics a molecule of adenosine and the other part of the structure mimics a molecule of tyrosine. So what does puromycin actually do? It interferes with the translation and causes premature release of the translated products. The mRNA display is an improvement over the ribosome display, in which the polypeptide under selection are attached to an enormous ribosomal RNA protein complex. This multiplies the chances of unpredictable interactions between the immobilized target and the ribosome and may lead to a loss of potential binders during the selection cycle. So the main advantage of mRNA display over the ribosomal display is that a covalent linking between the mRNA template and the peptide, and this covalent link reduces the loss of potential binder during the selection cycle. However, the main limitation of this is the synthesis of puromycin attached mRNA and the validation of peptide mRNA complex requires considerable efforts. The advantage of cell-free system over the include these can display very large libraries, that is up to 10 to the power 14 variants, and these are suitable for generating toxic peptides and allow the incorporation of modified amino acids at defined position to produce unique peptide inhibitors with proteolytic stability. These are the advantages of a cell-based system we have already studied. The fourth system is screening cyclic peptide libraries with E. coli. The split intin, the intin that we have studied before during the decoding the jargons, the split intin circular ligation of peptide and proteins, it is CILOPs, enable the generation of genetically encoded cyclic peptide libraries in bacteria. The peptides are encoded within an intin and the correct reading frame is maintained throughout the construct. After expression of the construct, the fusion protein folds to form an active intin and the peptide is circularized in vivo. In this way, a selection of active peptide is linked to cell growth by the disruption of the natural complex of the target rather than the complex formation, and this is known as reverse two hybrid system. Major advantage of this is the system is the in vivo screening of of a library of cyclic peptides. Molecules are more suitable to become good early drug leads. The limitation of this system is to screening peptides with only natural amino acids that are available. The drawback of Silope system is it is limited to library diversity of 10 to the power 8 variants, and the amount of information needed prior about the target, which is opposite to that of the fast display, in which the prior knowledge about the target was not required. Fifth is phenotypic screening of peptide libraries in mammalian cells. Here, using a pooled lentiviral system, they they express a motif intracellularly and screen it directly for their effect on cell proliferation. The lentivirus system can achieve stable and highly effective gene expression in a variety of mammalian cell types. Here, a deep sequencing on barcoded PCR products from cellular genomic DNA at different time points are assessed for the identification of active peptide. And using this approach, inhibitors were identified that drastically reduced the viability of pancreatic cancer lines. This is the main indication of the pancreatic cancer lines. This screen might provide new drug target and peptide drug leads, and it provides a rich data set covering phenotypes for the inhibition of thousands of interactions. The drawback of this screening method is it is limited to a library size of 10 to the power 6 variants, and there is a laborious validation steps to ratify both the peptide anti-proliferative activity and its targets. Sixth method is peptide arrays. The methodologies that we have studied till now mostly rely on tagging the peptide with a nucleotide label to allow a quick, easy, and accurate identification of the interesting peptide. But here, what we do is in this approach, there is synthesis of high purity peptide libraries on a solid surface, like glass or chip. So the techniques for parallel chemical synthesis of peptide on solid surface involve two. The first process is photolithographic synthesis, which is nothing but light-directed parallel chemical synthesis, and second is spot synthesis. In photolithographic synthesis, uses a photolabile protecting group to synthesize thousands of spots simultaneously, each with a unique peptide sequence. While in spot synthesis, peptides are synthesized by sequential spotting of small volumes of activated amino acid to a porous membrane. After synthesis, the peptide array is incubated with the target protein, and the complexes are decreased. And the complexes are detected using radioactive label, fluorescence, or antibodies. The advantage is that it provides a comprehensive information on binding peptides and quantitative comparison of affinity between peptides on the same chip. Other advantage is that there is a possibility to incorporate non-canonical amino acids, and this allows the investigation of interaction that rely on post-transcription modification. The application is epitope mapping. Epitope is a part of the antigen that binds to the antibody or paratope. Epitope mapping or identification of target protein by exposing an array enriched in known peptide motif to cell lysate is followed by identification of the protein using mass spectrometry. Also, there's a direct selection of peptides with antimicrobial activity. The main disadvantage of the peptide arrays is that relatively small size of the libraries, limited to 10 to the power 6 variants, and the rate of false positive and negative due to issues with the purity and quality of peptides on the chip. Second part of the webinar is optimization strategies. So next, the peptides have many advantages over drugs. As drugs over other molecules, they still suffer from poor pharmacokinetics. One common way to improve the pharmacological properties of a peptide drug candidate is to use it as the basis of an analog mimic, that is peptidomimetics, which involve manipulation of backbone and side chain atom to increase the bioavailability and biostability. Backbone modification involves changing amino functionality, C-alpha substitution, replacing the C-alpha H hydrogen with an R group, backbone extension, and carbonyl modification. While the sidechain modification vary from use of D-amino version of native residues to the introduction of huge arrays of non-canonical groups. So the first optimization strategy is backbone modification. Substituting an amino group for oxygen or sulfur creates decipeptides and thiodecipeptides respectively, and this is an well-established peptidomimetic approach. Decipeptides have been employed successfully as therapeutics, and the drug that is FDA approved by using backbone modification is romidapsin. Romidapsin is indicated for cutaneous T-cell lymphoma. Also, the thiodecipeptides have also appear in nature and have anti-tumor activity. Decipeptides alter backbone morphology and flexibility by reducing rotational barriers and thereby allow a greater range of conformational exploration. Other approach is methylation of the peptide nitrogen. The N-methylation constrains the backbone and removes the water molecule and thus can decrease binding-related entropy. Entropy is nothing but the lack of order in the binding. So the reported benefit due to this N-methylation is enhanced binding affinity, increased blood plasma half-life, and improved cell penetration. Apart from this, another approach is C-alpha manipulation. Replacement of the C-alpha with nitrogen alters backbone geometry significantly and changes the peptide's chemical properties. And these are known as azapeptides. These mimics are more resistant to enzymatic hydrolysis and are thus useful option for drug design. Azapeptides used in the clinical field include azetanavir, which is an antiretroviral used for the treatment of HIV. Azapeptide amino acids can be linked tandemly to form extended chains called azatides and combine with decipeptides to produce azadecipeptides. The C-alpha hydrogen can also be replaced by an R group to render single amino acid with two side chains, and this gives rise to a significantly subgroup of non-canonical amino acids that will be discussed in the next slide. Different peptidomimetic scaffolds can be constructed by inserting additional atoms into the backbone between the carboxyl and amino group. These additional atoms can be either carbon or heteroatoms such as oxygen and nitrogen. So there is a significant interest in this type of modification, mainly because this leads to the this backbone modification uh, provides resistance against the proteolytic degradation. Non-canonical sidechains. Most peptide therapeutics till now that we have studied use only canonical amino acids, that is the amino acids occurring in the nature, that is 20 amino acids found in the polypeptide synthesized by the ribosome. Introducing non-canonical amino acid greatly increases the sequence space and provides several benefits like selectivity, strong affinity, protease resistance against the hydrolysis, better cell penetration, and all this leads to enhanced potency of the drug. So till now, there are more than 200 commercially available non-canonical amino acids, and these range from simple methylated phenylalanine and more exotic functional group substitution and unusually large hydrophobic constructs. The core packing can also be optimized more effectively to improve the molecular stability. So the tremendous increase in sequence space does pose difficulty for experimental design owing to the constraints imposed by current library size, and this is the disadvantage. Other disadvantage of non-canonical sidechain modification is the cost. The cost of non-canonical amino acid is much more than the cost of non-canonical amino acid is more than compared to the canonical amino acids. While the computational techniques can help in exploration of the sequence space and the parameterization of non-canonical amino acids for computational protein design has made their incorporation more accessible, but the computational techniques are more laborious and a typical calculation can require up to 10,000 CPU hours for single residue mutation. And this is the main drawback. So D-amino acids. The simplest way to introduce non-canonical amino acid into a peptide is to exchange residue for their D-amino acid canonical amino acid stereoisomers. Almost all amino acids have chirality and therefore exist in two forms, dextrorotatory, that is D form, and levorotatory, L form. But in the nature, it is the L form that prevails. D peptides are not usually recognized by the L protein interaction partners, and this gives them a desirable property of being both non-immunogenic and they are resistant to proteolytic degradation. Also, better cell penetration and increased intracellular lifetime, and thus increases the potency by up to 5 times. Here, by simply exchanging L to D amino acid usually destroys the binding, as the side chain orientation with respect to the target are completely altered. The problem can be overcome by reversing the D peptide sequence, which restores the L amino acid side chain angles, and this is known as retroversion. Here, retroversion also often fails owing largely to the topographical differences in the helices. Here, we can see that the D peptides are mostly left-handed helices, while the L peptides are mostly right-handed helices, and the left-handed D helices remain left-handed even after the sequence is reversed, and the resulting topographic differences destroy the binding. The alternative approach for D peptide therapeutics is mirror synthesis. This involves synthesizing the target from D amino acid and using it as a bait for the randomized L amino peptide library. Successful hits are subsequently reconstructed with D amino acid, which bind to the native L protein target with a similar affinity as L to D form of the amino acid. Currently, these are limited. The limitation of D amino acid is currently these are limited to target D protein size of approximately 150 residues by commercial synthesis technique. And second limitation is if the target requires chaperone or obligate heterodimeric partner to fold, L chaperone are unlikely to recognize their D protein substrate because of their topographic is completely different. Now, the computational design represents an attractive alternative as most modeling packages are capable of handling D amino acids. Fourth optimization strategy is cyclic peptides. Cyclic peptides are polypeptide chains that can be formed by linking peptide termini to form a closed loop, and these links can be generated with an amide bond or other chemically stable bonds like lactone, ether, thioether, or disulfide bonds. The cyclization of arginyl glycyl aspartic acid, that is RGD peptide derivatives that are specific to particular integrin receptors, while the optimization of RGD peptides and optimization of ligand structure led to synthesis of cyclopentapeptide, that is RGD-FV. The clingitide that we have already discussed was derived from RGDFV using an N-methyl scan and is currently in the phase 2 clinical trials for the treatment of glioblastoma patients with an unmethylated MGMT gene promoter. While the several cyclic peptides found in nature that are used clinically include gramicidin, tyrosine, cyclosporin A, and vancomycin, and chitosan. And advantages of cyclic peptides include they are resistant to hydrolysis by endopeptidases and exopeptidases owing to the lack of both C and that is carbon and nitrogen termini. Because of the conformational rigidity, cyclic peptides display better biological activity compared to their linear equivalents. The rigid structure of the cyclic peptides decrease the entropy penalty, that is disorder in the bonding, and thereby enabling an enhanced binding and selectivity towards target molecule. While the disadvantages are the lack of flexibility, and second is a limited number of conformation may preclude adoption of the optimal conformation for binding of some targets, decreasing the affinity of these cyclic peptides compared to their linear equivalents. So, considering these both advantages and disadvantages, it can be said that cyclization is not therefore an universal solution for all protein peptide complexes. Another approach is the stippled peptides. Stippled peptides are alpha helical peptides that contain a synthetic hydrocarbon backbone linking various residues. Conformational stabilization introduced in one small stretch of a peptide can help to stabilize the entire structure. The two distinct conformational stabilization strategies that have been utilized to induce alpha helical structure are alpha-alpha disubstitution and macrocyclic bridge formation. These high levels of alpha helical contents are associated with following advantages, that is 5 to 5000 fold increase in the target affinity, strong reduction in proteolytic degradation, efficient cell penetration by endocytic vesicle tracking, extension of in vivo half-life, and specific disruption of protein-protein interactions in cultured cells. The disadvantages with this system include the increased hydrophobicity leads to decrease in the aqueous solubility. This decreased aqueous solubility makes full characterization difficult in biophysical assays as well as in vitro target interaction and activities assays. The structure-based design can help to increase the hydrophilicity of the staple peptides by replacing hydrophobic amino acids with non that are not expected to participate in interactions with the target with polar or charged residues. The ability of staple peptides to inhibit protein-protein interactions makes them attractive therapeutic candidate. Coming to the delivery strategies, to improve cell membrane permeability, proteolytic stability, and or target specificity, physicochemical modification of peptides uh, are used by addition of functional groups or by conjugation to polymers. The key approaches that are used for the delivery strategies include pegylation, lipidation, and the use of cell-penetrating peptides, while the other approaches to overcome peptide delivery problems are the use of carrier systems like colloidal delivery system, modified pathogens, endosome mechanism, or carbon nanotubes and missiles. So coming to the first delivery strategy, that is pegylation. Pegylation or covalent attachment of polyethylene glycol improves the stability, pharmacokinetics, and therapeutic activity of protein and peptide drugs, and this modifies various physicochemical properties such as molecular weight, size, solubility, and steric hindrance. Pegylation also reduces protein degradation by enabling interaction of protein with cell-associated receptor and enzymes or enabling specific cell-protein interactions. The pharmacokinetic and pharmacodynamic behavior of pegylated peptides may be affected by the type of linker that is present between the polyethylene glycol and peptide. The linkers such as phenyl, propyl, and amyl moiety have been used for the conjugation of C terminus, that is carbon terminus of protein, and polyethylene glycol. Phenyl linkers induce dense polyethylene glycol conformation, while both propyl and amyl linkers lend a loose conformation. The pegylation can mask the agent from the host immune system, that is decreases the immunogenicity and antigenicity and increases hydrodynamic size, that is size in the solution of the agent, which prolongs its circulatory time by decreasing renal excretion. It also provides water solubility to hydrophobic drugs and proteins. The example of pegylated drugs that are available in the market are pegfilgrastim, that is a pegylated recombinant human granulocyte colony-stimulating factor for the chemotherapy-induced neutropenia. Second is pegvisomant, that is a pegylated human growth hormone antagonist, which is used for acromegaly. Second is peptide lipidation. Fatty acid conjugates of polypeptide show improved transport across biological membranes, higher stability, and longer plasma half-life. The presence of lipid group in a peptide affects its secondary structure, hydrophobicity, and self-assembling propensity, and often conserves the ability to bind the target. Ditimer and liraglutide are myosylated and palmitoylated peptide analogs respectively and show strongly prolonged half-lives and slower biodegradation. Also, the attachment of fatty acids to ditimer and liraglutide enables them to cross the blood-brain barrier. Ditimer is a long-acting insulin, while the liraglutide is a long-acting glucagon-like peptide 1 agonist, which are used in diabetes. Also, the palmitylation improves cell permeability of peptides and improves their biological activity dramatically. It also stabilizes peptide tertiary structures and facilitates cellular delivery by increasing peptide lipid association. The disadvantages of peptide lipidation include a directly linked lipid polypeptide conjugate is likely insoluble in water, and the lipidized polypeptides in general tend to have low biological activity. Cell-penetrating peptides. Cell-penetrating peptides are relatively short peptides having less than 40 amino acids that can enter cell via different mechanisms, that is endocytosis, direct penetration, or reverse missile. Reverse missile will be discussed when we discuss the missiles. These are often used to assist the intracellular delivery of covalently and non-covalently conjugated bioactive cargoes. So, in recent years, many studies have revealed numerous applications of cell-penetrating peptides for the delivery of various cargoes like nucleic acid, polymers, liposomes, nanoparticles, and low molecular weight drugs. The cell-penetrating peptides can be divided into two categories depending on the origin. The first cell-penetrating peptides generated were truncated version of the full-length protein such as penetratin and TAT. The penetratin is developed from the naturally occurring Drosophila antennapedia transcription factor protein that has shown to internalize into the cells, and TAT is the strongly prolonged half-lives and slower biodegradation. Also, the attachment of fatty acids to determine liraglutide enables them to cross the blood-brain barrier. Determer is a long-acting insulin, while the liraglutide is a long-acting glucagon-like peptide 1 agonist, which are used in diabetes. Also, the palmitylation improves cell permeability of peptides and improves their biological activity dramatically. It also stabilizes peptide tertiary structures and facilitates cellular delivery by increasing peptide lipid association. The disadvantages of peptide lipidation include a directly linked lipid polypeptide conjugate is likely insoluble in water, and the lipidized polypeptides in general tend to have low biological activity. So the third delivery strategy that we are discussing were cell-penetrating peptides. Cell-penetrating peptides are relatively short peptides which contain less than 40 amino acids that can enter cells by different mechanisms, including endocytosis, direct penetration, or inverse missiles. And these are often used to assist in the intracellular delivery of covalently or non-covalently conjugated bioactive cargoes. In recent years, many studies have revealed numerous applications of cell-penetrating peptides for the delivery of various cargoes such as nucleic acid, polymers, liposomes, nanoparticles, and low molecular weight drugs. Cell-penetrating peptides can be divided into two categories on the basis of their origin. The first cell-penetrating peptides generated were truncated versions of full-length proteins like penetratin and TAT. The penetratin is developed from the naturally occurring Drosophila antennapedia transcription factor protein that has shown to internalize into the cells, while TAT is a transactivator of transcription is produced from the HIV virus that was first discovered to translocate into the cell membrane and enter the cells. While the combination of parts of several proteins can lead to new chimeric sequences that can have the cell-penetrating properties, and these are the second cell-penetrating peptides to be generated. The most widely used cell-penetrating peptides contain a chain of arginine or other positively charged amino acids like lysine and histidine. The first version of cell-penetrating peptides were composed of only natural amino acids, but more recently, to improve their properties, researchers have found it necessary to include non-primary and non-natural amino acids. Non-primary amino acids include phosphoserine, and non-natural amino acids include homolysine and homoarginine, which are formed by addition of methylene group to lysine and arginine respectively. The cellular uptake of cell-penetrating peptides depends highly on their secondary structure and is affected by the charge hydrogen bonds and helical properties of the peptide. Well, the cell-penetrating peptides have delivered small chemotherapeutic drugs like doxorubicin, methotrexate, cyclosporine, and paclitaxel. So next, one of the first bioactive cell-penetrating peptide discovered was a peptide derived from the N terminal, that is nitrogen terminal part of the tumor suppressor protein P14 ARF, and it showed pro-apoptotic activities. Well, recently they generated two intrinsically bioactive cell-penetrating peptide sequence called bioportraits that displayed potent anti-angiogenic activity in vivo. The next slide. The fourth delivery strategy is LFN peptide, that is lethal factor N peptide. Here the N means N terminal of N-terminal protective antigen binding domain of lethal factor. The anthrax lethal toxin uses the membrane-spanning translocase, that is protective antigen PA pore, to deliver the protein lethal factor LF from the endosome into the cytosol of the cell. So here you can see from the picture pore forming protein, that is PA83, which is above the receptor anthrax receptor ANTXR2. This pore forming protein PA83 interacts with the anthrax toxin receptor at the cell surface of the host and is then activated proteolytically by the fusin protease. As you can see, the fusin is there to generate a protein protective antigen PA63. PA63 then forms a heptameric or octameric prepore, and the lethal factor binds to the prepore through the 263 residue N-terminal domain with nanomolar affinity. The toxin complexes are internalized, as you can see, by the endocytosis via receptor-mediated endocytosis, in which the prepore is converted into a pore in the acidic environment of the endocytic vesicle. The protective antigen lethal factor N system transports naturally occurring or recombinantly expressed protein efficiently. This system has also been exploited for the delivery of antibody mimic natural peptides, cyclic peptides, enzymatic domains, and non-canonical amino acids fused to the C-terminus of the protective antigen binding domain of lethal factor into the cytosol via protective antigen. So the identification of cellular receptor for protective antigen and the related binary toxin will help in further understanding of the mechanism of action on cells and tissues. So coming to the colloidal delivery system. Colloidal delivery system has gained increasingly popularity in the contemporary world, and the colloidal carriers such as liposomes, microparticles, and nanoparticles have been investigated widely for the delivery of peptide therapeutics. The advantages of these carrier systems include reduced administration frequency, prolonged release of the drug, controlled plasma levels of the drug, and protection of the drug of sensitive proteins against the proteases. Liposomes. Liposomes are concentric bilayered vesicles in which an aqueous volume is enclosed entirely by a membranous lipid bilayer comprised mainly of natural or synthetic phospholipids in the presence or absence of cholesterol. They act as nanocarriers which are able to transport large variety of molecules such as drugs, small organic molecules, nucleic acids, metal complexes, or peptides. Different phospholipids are used in the preparation of liposomes, and they vary in length of the hydrocarbon chain and the number of unsaturated bonds. The most frequently used phospholipid for the preparation of peptide target liposomal drugs are composed of hydrogenated soy phosphatidylcholine, diasteroyl phosphatidylcholine, diasteroyl phosphatidylethanolamine, all of which contain two saturated or unsaturated alkyl chains of 18 carbon atoms each. The cholesterol is added to stabilize the membrane and obtain a fluidizing effect of the lipid bilayer or both. The liposomes are classified into unilamellar vesicles and multilamellar vesicles, while the unilamellar vesicles are also classified into large and small unilamellar vesicles. In unilamellar liposomes, vesicles have single phospholipid bilayer pairs enclosing an aqueous solution. The liposomes encapsulate enzymes that enter the cytoplasm or lysosomes of cells have been essential for the treatment of inherited diseases caused by the abnormal functioning of some intracellular enzymes, especially in the liver and the central nervous system cells. Liposomes have also been used to deliver beta-glucocerebrosidase inside the Kupffer cells in the liver, and modification of liposomes with mannose residue increased the capture because of the presence of mannose-specific receptor target cells. Liposomes have been used to encapsulate anti-tumor cells such as antibodies and peptides that target a particular type of tumor cells or tumor vasculature. Example of the drugs that use liposomal system include amphotericin B, anti-cancer drugs like doxorubicin and cytarabine, morphine, and insulin. Another method is microparticles. Microparticles are classified by the size of the particles ranging from 1 to 1000 micrometer in diameter. These are composed of biodegradable polymers which are used for systemic delivery of proteins and peptides, which include starch, alginate, collagen, polylactic glycolide, that is PLGA, and polycaprolide. They have got the advantage of providing controlled release of the drugs over several days to months. Because of its long safety history, that is PLGA has been studied extensively for protein and peptide delivery, and the various formulations that are available in the market include liposoline, triptozylin, octreotide, and lanzeotide. The biodegradable microspheres have been developed for attaining sustained release of recombinant interferon alpha 2b, which is used for the treatment of hepatitis C virus infection and some form of cancer. The major advantage of microspheres is their provision of stability for labile compounds that are easily degraded in the intestinal epithelium. However, it is found that large size of the microparticles can have a negative impact on the efficacy of the peptide drug, and thus small-scale delivery system might be more suitable and effective in the future. Nanoparticles. Nanoparticles are colloidal carriers that range in the size from 10 to 1000 nanometer. They are composed of materials such as polymers, lipids, and dendrimers, and have been used for target delivery of small molecule drugs. The size, surface charge, and nature of encapsulated nanoparticles influence the absorption and delivery of the molecule. Various natural and synthetic polymers like PLGA, polymethyl methacrylate, polylactic acid, that is PLA, alginate, cyclodextrin, and chitosan and its derivative have been used for the production of nanoparticles. The use of nanotechnology system at the surface of liposome has led to the development of lipid nanoparticles, which are superior, which have some superior properties as compared to the liposomes. This system increases protein encapsulation and provides protection against enzymatic degradation. Glycol chitosan nanoparticles also display fast cellular and tissue internalization into the tumor. Also, chitosan and its derivative have been reported to be biodegradable, biocompatible, and poorly immunogenic. Nanoparticles are used for the delivery of a combination of small molecule transforming growth factor beta inhibitors and immunostimulant interleukin 2. While nanotechnology has promising avenues for peptide therapeutics, some challenges remain with regard to the low protein and peptide incorporation efficiency, uncontrolled molecule release, susceptibility to protein aggregation, and possible accumulation of non-biodegradable polymers in the tissue. Carbon nanotubes. Carbon nanotubes are hollow, cylindrical nanostructures consisting of hexagonal arrangement of sp2 hybridized carbon atoms. These are divided into two broad categories, namely single-walled carbon nanotubes and multi-walled carbon nanotubes, depending on the number of sheets rolled into concentric cylinders. These are characterized by high electrical and thermal conductivity, great strength, and rigidity. The advantages of carbon nanotubes are large surface area, functionalization of ligands and molecules, and the ability to internalize into a wide variety of cells. But the limitation of these carbon nanotubes is that it can be difficult to solubilize in aqueous solutions. These are found by self-assembly of amphiphilic molecules and in general contain an inner hydrophobic core and outer hydrophilic corona, while the inverse or inverted missiles have the core is found by a hydrophilic part and the outer shell is found by hydrophobic part. So the advantages include they are able to encapsulate macromolecules in their core, ensure chemical and physical stability of encapsulated macromolecules, ensure conservation of peptide structure, and provide sustained release of encapsulated drugs. And thus, they have improved drug pharmacokinetic profile. A study showed that peptide such as glucagon-like peptide 1, pancreatic polypeptide, and neuropeptide Y form large heterogeneous aggregate in aqueous media. But this was prevented when the peptides were incubated with sterically stabilized micelles. Peptides associated with micelles exhibit enhanced therapeutic efficacy owing to the higher availability of peptides in stable and active form at the target site, and this was demonstrated using GLP-1 sterically stabilized phospholipid nanomicelles, vasoactive intestinal peptide sterically stabilized phospholipid nanomicelles. However, these are relatively safe and materials are biocompatible and biodegradable. Finally, on a concluding note, precise modulation of specific protein-peptide interaction is a promising new therapeutic strategy. Employing peptides as a means of modulation represents an exciting path towards this goal. Many different technologies have been introduced to aid in discovery of peptide binders suitable to become framework for future drugs, and these different technologies have got both advantages and their own limitations. Future for peptide drug looks increasingly bright as more candidates are now approved or are in the clinical trials. Thank you. This was all from my side. Over to Moti sir.
Sonali and Dr. Shashikiran is there in addition to me and you. So thank you very much. Do you have any questions? Okay, so if no further questions are there, first of all, we would like to thank Dr. Vikas Sharma for his excellent presentation. And this presentation has been recorded already, and this will be, made available for other, participants